Bismillahirrahmanirrahim. Assalamu alaikum everyone. My name is Rima Shaheen and I'm affiliated with the Ministry of Higher Education, Saudi Arabia. Today I'm presenting our paper at ACDSA 2026 and IEEE conference titled A Genetic Algorithm for Fast and Accurate Error Correction in Genome Sequence Assembly. This research is a collaborative work. The paper is led by Dr. Shakil Ahmed as a principal author, along with a team of co authors from different institutions. Firstly, I will briefly introduce a problem, the review related work, then I will explain our methodology, key results, conclude with the future directions, inshallah. Let's start with the introduction. Next generation sequencing technologies generate the large volume of genomic reads at high speed. However, these reads often contain base level errors that negatively affect genome's assembly quality. Therefore, the accurate and efficient error corrections is a critical pre-processing step in the genome data analysis. Let's move toward to know about the problem statement. NGS reads commonly contain the errors such as substitutions, insertions, and deletions. Existing error correction methods often require the high computation and memory, struggle to scale the large data sets, and ensure reduced accuracy across different sequencing platforms. Therefore, there is a clear need of an error correction method that provides the high accuracy while reducing the execution time. From just a practical perspective, our approach improves the genome assembly quality, reduce execution time, and can be applied across the multiple sequencing platforms such as Illumina, PacBio, and HiFi and Oxford Nanopore. From a theoretical perspective, this work shows that genetic algorithms are effective for the large bioinformatic problems and also offer a strong alternatives to supervised machine learning for error correction. The main aim of this research is to design a fast and accurate error correction algorithm for genome sequence assembly using a genetic algorithm approach. Unlike supervised methods, our approach does not rely on the label training data and focus on the correcting the substitutions error efficiency. We proposed, which we are abbreviating as the FAGSA, and evaluate it on multiple real world datasets where it achieves higher accuracy and the faster execution time compared to existing tools while also addressing scalability challenges. So, our methodology is based on a genetic algorithm driven error correction model called FAGSA, which we are just pronouncing for the abbreviation. We treat genome error correction as a combinational optimization problem and also use evolutionary operations such as the selection, crossover, and mutation of find optimal base corrections. This model is designed to specifically for the substitutions errors and does not require label training data. We evaluate the method using the benchmark datasets and measure performance using accuracy, precision, recall, F1 score, then also execution time and the memory usage as well. So our experiments, we use 13 benchmarks genome datasets collected from Illumina, PacBio, HiFi, and Oxford Nanopore platforms. These datasets include different genome types, read lengths, coverage levels, and error profiles. In the proposed framework, raw sequencing reads are first analyzed to detect just error based positions, And then a genetic algorithm is then initialized with the random candidate corrections, and fitness is evaluated by the minimizing base mismatches. Through this selection, crossover, and mutation, and the algorithm evolves improved solutions and output corrected genome reads with higher accuracy. In this study, the algorithm were evaluated using benchmark dataset with the different genome lengths, read lengths, and number of the reads. We compare accuracy and execution time using the standard matrix measured as a seconds per megabyte. All experiments were conducted on the same hardware environment to ensure fair comparison. The results show that this algorithm or this study algorithm achieved consistency, higher accuracy, and the faster execution time compared to the existing methods across most datasets. While some algorithms failed on a certain dataset due to memory or a data behavior issue, so FAGSA remained stable and effective. However, the current version of proposed algorithm focuses on the substitutions error, and the future work will be extend it to handle the insertion and deletion errors as well. So if I sum up all the key findings here, to summarize the key findings, the proposed algorithm, that is the FAGSA consistency, achieve higher accuracy and the faster execution compared to existing error correction methods. The approach generalized well across a different sequencing platform and the dataset size showing stable performance, while memory usage increased for a very large dataset, and also highlight clear direction for scalability improvement in the future work. These results provide the important insight into the effectiveness of genetic algorithm based on optimization for genome error corrections. So the proposed model in proposed algorithm, you can say that shows the strong performance in the term of accuracy and execution time across different stat sets and sequencing platforms as well. From a just practical perspective, the method can be applied in the real-world genome assembly pipelines without relying on label training data. However, the current study is limited to substitutions errors and the memory just use of the memory increasing for the every large data set. So let's conclude this all proposed study, that in conclusion what we can say that the study proposed a genetic algorithm based on approach for the error correction in genome sequence assembly. The method focus on substitution error correction using the evolutionary operations such as the selection, crossover, and mutation. On our result shows that the proposed algorithm achieves higher accuracy and significantly reduce the execution time compared to existing methods. The approach performs consistently across different genome size, read lengths, and the sequencing platforms, demonstrating the effectiveness of evolutionary optimization for large genome problems. As a future work, we plan to extend this algorithm to handle insertion and deletion errors in addition to substitution errors. We also aim to reduce the memory usage through compact encoding and streaming based on processing for very large data set. Further optimization of genetic parameters will be explored to improve the scalability and reduce the execution time while maintaining accuracy. Thank you for your time and attention. And if you want to ask me any question, you can ask.